Hello students, myself Ms. Jeevithaa, Assistant Professor, Department of Neuroscience Technology, School of Allied Health Sciences, Naiga Mission Research Foundation, deemed to be University, Salem. Today we are going to have a topic, development of nervous system. The nervous system is highly organized network that detects stimuli, process informations and coordinates responses through electrical and the chemical signaling. Structurally, it is divided into central nervous system and the peripheral nervous system. Central nervous system comprises of brain and the spinal cord. Thus, peripheral nervous system, which comprises of cranial nerves, peripheral nerves and the ganglions, which is peripheral ganglions. Further, this functionally divided into somatic nervous system and the autonomic nervous system. This somatic nervous system, which is responsible for the voluntary movements, and the autonomic nervous system, which is responsible for the involuntary movements, which is not under our control. This ANS, autonomic nervous system, includes sympathetic and the parasympathetic, and the also it comprises of enteric components, that is gut-brain axis, also called as enteric nervous system. This is the graphical mapping of nervous system. By this way, we can have the classification of nervous system. This nervous system comprises of, as we discussed before, this peripheral nervous system and the central nervous system. This peripheral nervous system further classified into autonomic nervous system and the somatic nervous system. This autonomic nervous system further classified into sympathetic, parasympathetic and the enteric nervous system. This enteric nervous system also called as gut-brain axis, the communication between the gut and the brain. Initially, our nervous system is developed from the germ layers. Embryologically, we can have a close note on this. Almost the entire nervous system is derived from the ectoderm. The germ layer of this is comprises of 3 layers. Initially, it is started with ectoderm, mesoderm and the endoderm. Before the gastrulation part, we can have a bilaminar disc. In that bilaminar disc, we can have a 2 layers. One is hypoblast, another one is epiblast. Okay. This 3 related derivatives, neural tube, neural crust cells and the cranial ectoderm placodes. Week 3 of the intrauterine life, we will have a gastrulation process. In this gastrulation process, the 3 trilaminar disc will be obtained or processed. During that stage, the ectoderm, mesoderm, endoderm gets developed. The notochord, which is the mid part of the mesoderm, which is further developed, and the pericardial plate form in the midline mesoderm beneath the dorsal ectoderm. We can see the initial layer is ectoderm. This ectoderm is classified into neuroectoderm and the surface ectoderm. This neuroectoderm developed to form the CNS, central nervous system. The surface ectoderm, which is developed to form the placodes. Signals from notochord induce overlying the ectoderm to become neuroectoderm, forming the neural plate. The lateral ectoderm remains surface ectoderm, later forming the epidermis and its appendages. Appendages includes sweat glands, mammary glands and the sebaceous glands, otic epithelial cells and etc. As we discussed before, neural plate, neural groove and neural fold are the initial stage of the development of this nervous system. By this way, neural plate is first appears in the cranial region about 18th to 19th day of a intrauterine life, and it extends to the caudally to form the tail part. The central region of the plate invaginates, that it gets folded inverted to form a neural groove. This neural groove, it's get attached. As we see in this picture, this neural plate invaginate to form a neural groove. This neural groove further gets attached to form a tube-like structure. This is neuropore. Later, the neural crest of this two hump or the folding become a somites of peripheral nervous system. These somites get developed to form a dorsal root ganglions. Lateral margins elevated to become neural folds on either side of the group. This neural plate border between plate and surface ectoderm is source of future neural crest cells. This neurulation and the neuropore closure. Neurulation is the process later occurs next to the gastrulation. Gastrulation occurs on the 3rd week of the intrauterine life, and this neurulation occurs on the 5th week of the intrauterine life. Okay, neurulation is the process by which the whole CNS and the peripheral nervous system is going to be developed. In this, the neural folds move towards each other and fuse to form a cervical region, and this the fused possessed cranially and caudally to form the neural plate into a closed neural tube. You can see in this picture that it has two pores, two openings. The head portion is called as cranial pore and the bottom portion is called as caudal pore. Okay. The cranially, that is anteriorly, the neuropore closes around the 25th day of intrauterine life, and the caudally part, which is a posterior neuropore, closes around a day of 27 to 28th day. Okay, the proper closure is essential because this leads to the failure of this neural pore closure leads to neural tube defects. Primary and secondary neurulation. Primary neurulation. In the initial stage of the neural fold formation. The primary neurulation, the folding and the closure of neural plate to form most of the brain and the spinal cord. The caudal cell mass in the tail region later cavitates and fuses with the primary tube. That is the process of secondary neurulation occurs. This secondary neurulation forms the most caudal spinal cord segments and the filum terminae. Filum terminae is the last part of the spinal cord, at most last part of the spinal cord. This closure of caudal pore which leads to abnormalities like spinal occulta or meningeocele, myelomeningeocele and tethered cord syndrome. Early neural tube layers. Initially, the neural tube wall is made of pseudostratified neuroepithelial cells. This ventricular, that is ependymal zone of this spinal cord development, which is get proliferate, that is got multiplied to form a neuroepithelial stem cells. That adjacent to the lumen. Lumen is a cavity which is presented here. Okay, these are the cells, ependymal cells, which is presented. Okay, and then the next layer of this ependymal layer, mantle layer. This mantle layer has a neuroblast that's migrating toward to form a further grey matter, and that next layer has a marginal layer. This marginal layer, it is a peripheral zone of axons, later become a white matter with myelination. This picture explains the neural tube formation. This neural tube formation has a cavity and the cavity is also called as lumen. This lumen has two sections, one is roof plate, another one is floor plate. Okay, which is divided into two parts, that is alar lamina, basal lamina by a imaginary line, that is sulcus limitans. Okay, the dorsoventral organization, alar and the basal plate. As we said before, this sulcus limitans divides the spinal cord into alar plate and the basal plate. Longitudinal sulcus limitans appears in the lateral wall of neural tube. Dorsal to sulcus alar plate and the ventral to sulcus is a your basal plate. This alar plate giving rise to the afferent neurofibers. Afferent neurofibers which is responsible for the sensory neuronal columns, and the basal plate which forms the motor neurons. This motor neurons forming motor neurons which is presented on the neuronal columns. Midline roof and the floor plates act as a key signaling center rather than the neuronal zones. The molecular control of the tube patterning. There are some chemical components or the molecular components which is responsible for the development. Initially we can have a SHH, that is sonic hedgehog from notochord, and the floor plate induces ventral, that is our basal plate, neuronal fades. This BMP protein, that is bone morphogenetic protein, and the related signals form roof plate and the overlying ectoderm specific dorsal interneurons. Dorsal is always a alar plate and the ventral is always a basal plate. The distinct combination of transcriptions factor define progenitor domains for different neuron cells. This rostrocaudal identity is patterned by the Hox genes and the morphogen gradient such as retinoic acid and the FGF. FGF is a growth factor of fibroblast. These are the main molecular components which is responsible in the development of neural tube or neural tube patterning. Brain flexures and the primary vesicles. The cranial neurotube enlarges and bends at cephalic and central and cervical flexure. This cephalic flexure is presented at the midbrain region and the cervical flexure which is presented at the hindbrain region. The three primary brain vesicles are prosencephalon, mesencephalon and the rhombencephalon area. This prosencephalon is also called as forebrain. This mesencephalon is also called as midbrain and the rhombencephalon is also called as hindbrain. Okay, and then the cervical flexure demarcates the hindbrain from the spinal cord. That is the cervical flexure which separates or differentiate the hindbrain from the spinal cord region, and then the cephalic flexure which demarcates or differentiate the midbrain from the forebrain. The later pontine flexure influences between fourth ventricle and the cerebellum. The later pontine flexure I have said, right? This pontine flexure which influences the development of fourth ventricle which is presented at the brainstem region. This brainstem region has our cerebellum, secondary brain vesicles and the derivatives. Prosencephalon divides into telencephalon and diencephalon. As you can see this, the three primary vesicles which is developed in the fourth week of the intrauterine life or embryonic stage, and this 5 secondary brain vesicles which is developed on the fifth week of the embryological stage or intrauterine life. This prosencephalon, that is our forebrain, is developed to form a telencephalon and the diencephalon. This mesencephalon does not differentiate it is developed as mesencephalon, and the rhombencephalon, that is hindbrain region, which is developed to form a metencephalon and the myelencephalon. This mesencephalon remains single and becomes midbrain to form a tectum, tegmentum, cerebral peduncles, and the rhombencephalon divides into the metencephalon and the myencephalon. This further developed to form the pons, cerebellum, and the myelencephalon is developed to form the medulla oblongata. Okay, this each vesicle surrounds a part of future ventricular system. The ventricular system and the CSF formation. CSF is a cerebrospinal fluid formation. The telencephalon which encloses the paired lateral ventricles. Diencephalon surrounds the third ventricle. Okay, and then this mesencephalon narrows to form a cerebral aqueduct which paves a way for the draining of or a pathway for the CSF which is formed on the choroid plexus region. This mesencephalon and the myelencephalon form the roof of and also the floor of the fourth ventricle. The choroid plexus arises from the ependyma and the vascular mesenchyme in ventricular rows to produce cerebrospinal fluid. Okay, this is our lateral ventricle. This is fourth ventricle. Interventricular foramen. By this way, it reaches the fourth ventricle and drains out. If there is any obstruction in this drainage, it leads to hydrocephalus. Hydrocephalus is further classified into two. One is communicating, another one is non-communicating. Based upon the function, it is classified. The development of spinal cord. The spinal cord develops from the neural tube caudal to the further fourth somite level. The ventricular zone cells generate neuroblast for the dorsal, that is sensory, and the ventral, that is motor gray horns. The axons forms these neurons grow into marginal layer forming ascending and the descending tracts of spinal cord. As development proceeds, white matter thickens around the central grey matter. This is grey matter and this one is your white matter. Ascent of spinal cord and the formation of cauda equina. In fetus, the spinal cord extends nearly the full length of the vertebral canal. The vertebral column grows faster than the cord, so the cord ends progressively higher. But in the adult stage, the conus medullaris, which is ends around the L1 to L2 region, the segment of lumbar 1 and the lumbar 2. The lumbar and the sacral roots lengthen downwards as a cauda equina, which is a horsetail part of multiple nerve fibers. As we know, the spinal cord is covered by a three layer, that is meningeal layer is pia, arachnoid and the dura mater, which is a meningeal layer or a covering. Okay. This pia, which is derived phylum terminale, anchors the distal cord to the coccygeal part. Neural crest, origin, EMT and the migration. This neural crest, we have seen this neural crest in the development of the neural foldings. During the neural fold, we have lifted the somites. Okay, these somites are developed from the neural crest. This neural crest cells forms at the neural plate border and the dorsal neural tube during neurulation process. Undergo epithelial mesenchymal transmissions and the delaminate from the dorsal tube. Migrate along cranial, cardiac, trunk, vagal and sacral pathways to diverse destinations. Neural crest cells are multipotent, giving rise to neuronal glial and the non-neuronal tissues. This neural crest cells, which is developed or differentiated to form a melanocytes, peripheral neurons, chromaffin cells, Schwann glial cells, smooth muscle cells and the cartilages and bones. Neural crest cells, derivation of peripheral nervous system. The neural crest cells, which is developed to form a dorsal root ganglion, which has a vital role in the transferring the neural information or the sensory information from the skin to the thalamus, higher order. Sensory neurons of a dorsal root, spinal ganglia and the associated satellite cells. Satellite cells are the glial cells. This major portion of the cranial sensory ganglia, that is trigeminal, facial, hypoglossal and vagus, and the Schwann cells that myelinate and support all peripheral nerves. Additionally, derivatives include adrenal medulla, chromaffin cells, melanocytes and such craniofacial mesenchymes. Ectodermal placodes. These ectodermal placodes are developed from the surface ectoderm. Cranial ectodermal placodes are localized at thickening of head ectoderm. You can see this otic placode, nasal placode. This otic placode is developed to form a inner ear, that is our cochlear part. The nasal placode, which is developed to form the olfactory epithelium. Also the lens placode, which is developed to form a lens of the eye. The epibranchial placodes contribute neurons to cranial sensory ganglions of the cranial of 7, 9 and the 10. 7 is your facial and this 10th is your vagus. Many cranial sensory ganglia contain mixture of crest derivative and placode derived neurons. The development of PNS, peripheral nervous system. The somatic motor axons emerges from the ventral horn and exits via the ventral roots. This spinal cord has a two part. One is sensory, another one is your motor. Sensory part and this is your motor part, which is presented on the dorsal side. This motor is presented on the ventral side. This afferent and efferent nerve fibers conjointly to form a spinal root formation. Spinal root formation. This spinal root formation is it is exited by the intervertebral foramen. Further it is called as white matter ramus, dorsal ramus and the ventral ramus. Okay. The sensory neurons in the dorsal root ganglion send central axons into the dorsal horn and the peripheral branches to the receptors. The ventral and the dorsal roots join to form a mixed cranial nerves and then divided into rami, that is dorsal rami and the ventral rami, and also plexus, that is brachial plexus and the lumbosacral plexus. At the shoulder portion we can see the brachial plexus. At the hip portion we can see a lumbosacral plexus. Plexus is a group of nerve fibers which is emerging out. The Schwann cells ensheath and myelinate large peripheral axons enabling fast conductions. Okay. Then autonomic nervous system. This autonomic nervous system provides a involuntary control of smooth muscles, cardiac muscles and the glands. Central autonomic centers, that is hypothalamus, brainstems, nuclei, spinal intermediolateral cell column, which arises from your neural tube. Peripheral autonomic ganglia and the postganglionic neurons largely derive from the neural crest. Functional pathways consist of preganglionic neurons in CNS and the postganglionic neurons in the peripheral ganglia. Development of sympathetic nervous system. This sympathetic nervous system, which is responsible for the fight and flight response. Trunk neural crest cells migrate ventrolaterally to regions near dorsal aorta. Aggregation of these cells form paravertebral sympathetic chain ganglia along vertebral column. Some crest cells condense anterior to aorta to form prevertebral, that is pre-aortic ganglia. Example, celiac and the mesenteric. Others invade adrenal primordium to form a adrenal medulla. Chromaffin cells, preganglionic neurons arises in the thoracolumbar spinal cord. Development of parasympathetic and enteric system. The enteric system is responsible for the gut and the brain communication, that is gut-brain axis. Okay. This parasympathetic nervous system, that is responsible for the rest and digestion. The cranial neural crest cells form parasympathetic ganglia associated with the cranial nerves, that is ciliary ganglion, ptericopalatal ganglion, submandibular ganglion and the otic ganglion. The vagal and the sacral neural crest cells migrate into the gut wall to form a enteric ganglia. The enteric neurons organizes into mesenteric, that is orboch and the submucosal, that is Meissner plexus. The preganglionic parasympathetic neurons originated in specific brainstem nuclei and the sacral cord segment. ENS, that is enteric nervous system, it acts as a mini brain in the gut with extensive local reflex circuits. You can see this image, the enteric nervous system which has a walls. This is a mucosal plexus, that is Meissner's plexus. This one is a mesenteric or orboch plexus, which is composed of ganglias. Neurogenesis, gliogenesis. The neurogenesis is the formation of neurons or neurocells. Gliogenesis is the process by which the glial cells are produced or formed. The neural stem cells in the ventricular zone sequentially generate neuroblast, that is neurons, and the glial cells. Glioblast. Glioblast includes astrocytes and the oligodendrocyte cells. Neurons migrate along radial glial fibers to establish cortical layers and the neural groups. Tangential migrations contribute interneurons to cortex and the brainstem. Accurate migration is vital. Disturbances causes malformations like lesioncephaly and heterotophia. These stem cells act as a progenitor cells which is developed to form these neurons, astrocytes and the oligodendrocytes. The formation of neuron is named as neurogenesis, and this formation of glial cells are called as gliogenesis. Axon guidances and the synaptogenesis. The growth cones responded to attractive and the repulsive cues. Example, netrins, semaphorins and ephrins in the extracellular environment. Cell adhesion molecules and the guidance cues steer axons to appropriate targets. When axons reach targets, synapses form. Competition and activity refine connections. Experience dependent synaptic stabilization and the pruning shape. Mature circuits. You can see this synapse get formed. These are the arborization of or dendrites, which is get a stabilization, that is synaptic stabilization. Myelination and the postnatal maturation. CNS myelination by oligodendrocytes begins late in the gestation and continues into adolescence. PNS myelination by Schwann cells improves conduction velocity in peripheral nerves. Different tracts myelinate at different times, correlating with the development milestones. Ongoing plasticity allows experience to shape neural connectivity, especially in the early life. Development disorder. Finally, we are getting into a topic that is the development disorder. This development disorder which explains the defects which is occurring during the development stage of nervous system. If there any error occurs during the development, the central nervous system has a defect, that is neural tube defects. Initially, we have gone through the topic which is cranial pore and the caudal pore. This cranial pore should be enclosed at the day of 25, and this caudal pore should be enclosed at the stage of 27th. If there are any defects of closure of this cranial neuropore, that leads to anencephaly, encephalocele, and this caudal pore which is causes any defects to closure, that leads to spina bifida, spina occulta, meningomyelocele, okay. And then this hydrocephalus is another condition and also it has cortical malformations results from failed neurulations or the neuropore closures. This hydrocephalus arises from the disturbed cerebrospinal fluid flow or absorption, which is leading to ventricular enlargement. Anencephaly, encephalocele, spina bifida occulta, meningeocele and meningomyelocele, closed spinal dysplasium. This anencephaly, which has a open brain and the lack of skull vault, and here this encephaly, which has a herniation of the meninges, which is a covering of your brain region. This spina bifida occulta, which is a closed asymptomatic in which some of the vertebrae are not completely closed. The closed spinal dysraphism deficiency of least 2 vertebral arches here covered with the lymphoma. Meningeocele. Meningeocele is the protrusion of the meninges filled with CSF through defects in the skull or spine. Myelomeningocele, which is similar to the meningeocele, but it additionally has this meningeal layer too. The peripheral nervous system and the autonomic nervous system defects. The neurocrystopathies. Neurocrystopathy are a group of disease that caused by the abnormal generation, migration or differentiation of neural crest cells. You can see a condition which is a very rare genetical condition is Hirschsprung's colon. This Hirschsprung's colon disease, you can see the swollen colon with the shrunken rectum. It is a very rare genetical disorder. And the familial dysautonomia, craniofacial anomalies reflect abnormal neural crest development. The crest derived tumors, that is neuroblastoma, pheochromocytoma, paraganglioma. Many conditions reflects failure of closure, migration, differentiation or myelination. Here we can see a adrenal part. In this adrenal part of the kidney, we can have a hump like structure, right? This leads to extra production of adrenaline. That condition is called as pheochromocytoma. Thank you children for watching my video.